So I'm going to talk about the connection of synaptic plasticity to our immune system, how that might contribute to processes of neurodegeneration as they are involved in Alzheimer's disease. And actually the idea that the nervous system and the immune system might have something in common is not actually a new one, but here this is a curiosity that in the 1960s people were looking for mechanisms how the brain can store information in the long run, and then neuroscientists had no conceptual idea how that could be done, and they were looking at the immunologists how such processes could work, and there it was found in immunology that actually antibodies, or B-cell producing antibodies, can memorize a parasite and bacteria that has entered the body before. So there was something in the surface of these antibodies that had a memory about a parasite that has tried to invade our body, and then there's a better immune protection if that is done. Neuroscientists at that time thought, hey, that might be a cool idea for the nervous system as well. So what they did is, actually crazy experiment, they trained rats to avoid the dark, which rats usually don't do. They would go into the dark and avoid the open space, but when they went into the dark they got an electric shock, so they learned to avoid the dark. Didn't do them any good because they were killed right away, and the brain of these rats were fed to other brains who have been naive to the situation, and actually these rats learned faster in the end. So the interpretation came then in the New York Times at the headline that they made. They said, well, if that is true, you can actually eat your professors instead of doing stupid learning sessions, because then you can eat proteins of molecules. As we know now, it's not as easy. They're not single molecules that store information. It's much more complicated, but it shows that there's a long-term history between the immune system and the nervous system. And here you see how closely they interact in the human brain. Here you see in red the immune cells in the brain. They are called microglia cells. They are a subtype of glia cells, and they are actually very close to synaptic sites, and they can be involved either in shaping synapses, they can actually prune synapses, or by releasing neurotrophic factors, they can actually promote synapse formation in many senses. So also during development and in our brain, there's a huge interaction between immune cells and neurons. And keep in mind that 10% of all cells in the brain are microglia cells, so they are really a very important component. And one of the molecules we studied for many, many years is the brain-derived neurotrophic factor, which can be released by neurons' astrocytes, but it can also be released by microglia cells under certain conditions which are important for the development of the brain. It might promote synaptic plasticity, and the reduction in BDNF expression might actually be bad news if the immune system is activated. So actually we wanted to know first about the role of BDNF in microglia research. So we looked at the gene expression of BDNF in microglia, and you see it's a lot lower than it is in neurons. We thought, well, it might be negligible, so we might not have to contribute and have to take it into account. But if you look more closely, that you can stimulate microglia cells with bacterial cell wall components, which will also play a role later in my talk. It's the LPS, the lipopolysaccharide, that's the main component of bacterial cell walls, like from Salmonella. And this LPS actually leads to a lower reduction of BDNF in microglia cells, which, as I said before, might actually be bad news for neurons, because then the neuroprotective molecules are released to a lesser extent, and that's why microglia cells might be important. Here you also see, you have heard before, that you can actually apply A-beta to organotypic cultures, as Ed Koo has told you before. We can do the same here, and if you can see here that the LPS application together with A-beta. So if you have an infection and you add A-beta, that the BDNF production is dramatically reduced, as one can also see in Alzheimer's patients, that there also the BDNF level is heavily reduced. So that might be another indication of an interesting connection. One of the receptors for all neurotrophins is the P75 Neurotrophian Receptor. So we have knockout mice and we studied these receptors very carefully, but here it comes into play because the P75 Neurotrophian Receptor also binds A-beta in the nanomolar range. So most likely it's a physiologically relevant component of how A-beta might actually add and kill cells in neurons. And also here the P75 also binds the oligomeric versions of A-beta, not just the soluble molecules. And as you can see here, if we remove P75 in the mouse genome from neurons, that it will be heavily affected and it will actually rescue the LTP defects that have been shown before by Ed Koo and by Dr. Sachikuma. So as you can see here, it's again an LTP experiment. You see here there's a high frequency stimulation and you see the lower LTP graphs for a mouse model of Alzheimer's disease, the ABPPS1 mice, which are knock-in mice of human mutations related to Alzheimer's disease. And if we knock out as well in these mice the P75, the LTP is normal again, which again shows you that probably one of the mediators of negative effects on synaptic plasticity might be P75, because the P75 neurotrophian receptor opposes the other neurotrophian receptor, the TrkB receptor, which has mostly positive effects on neuronal survival and synaptic plasticity. And for P75, it's quite the opposite. So if this receptor is activated, it might actually be bad news for neuronal survival. But not only synaptic plasticity, so functional aspects of transmission are affected. Also, when we look at substructures of neurons, you also have seen pictures like that in Ed Koo's talk. We count how many of these spines we see. We call it then spine density. We measure the length and we see the head size, which is an indicator, also a structural indicator of the strength of synapses, of the size of the postsynaptic density. And also here, to cut a long story short, we see that the P75 knockout mice are protected from all the bad effects that A-beta has on the structure of these neurons, where you see spine loss, they become longer, thinner, they lose spine head, and all that is corrected if the P75 is taken out of the genome. And also you can see that the overall morphology of neurons is rescued if the ABPPS1 mice are crossed to the P75 knockout mice. You see that there's not a reduction in complexity anymore. They have the same dendritic complexity as control mice, which is another indicator. The amyloid beta treatment actually leads to structural alteration that actually then are leading to less spines and to smaller synaptic connections, in a way that the spines lose volume, and that can all be rescued in P75 knockout mice as well. The signaling goes via probably one of the signaling molecules, the actin-binding proteins. It's the ROC molecule. So probably A-beta, we are P75 is destabilizing the actin cytoskeleton because, as you might know, there's a constant turnover of actin monomers added to actin filaments, and when this equilibrium turns to the negative side, actually the actin filaments become more unstable and you lose structural components, which is known to be contributed by P75. And if A-beta is activating P75, it might actually be bad news for the stability of synaptic connection, and that's precisely what you see in the early times of the initiation of Alzheimer's disease. It has also been shown that P75 is upregulated around plaques to a high extent and BDNF is downregulated. So something that is neuroprotective is downregulated, and something that can lead to cell death or at least to negative synaptic plasticity is upregulated. So in the moment we are studying microglia cells under these conditions in APP-PS1 mice or in P75 knockout mice, and as you will see also under other conditions. Here you see organotypic cultures, and you see in these cultures how active microglia cells are. They are as active as they are in the brain. They are constantly monitoring their environment. They are housekeepers, but they can turn into soldiers when they become activated. And using confocal and two-photon imaging, we can actually study these microglia cells in detail. And when these cells become activated, they lose their processes. They basically just keep their cell body move around, and they move to the side where they think an immunological reaction is happening, and then might fight it. It can be a pathogen, but it can at the same time also be a neuron that might be affected by the activation of microglia cells. So it's also to all the talks you have heard this morning about A-beta and tau and caspases, it might also be important to keep in mind that 10% of the cells in our brain are microglia cells, and the activation of microglia cells might not just play a role in the progression of Alzheimer's disease in the very end, it might actually promote the progression of the disease, or actually the origin of the disease might actually be at least partly be caused by microglia cells, because as Ed Kuh has highlighted in his talk, there might be many roads to destruction, and one of the roads might actually be paved with the activation of microglia cells, among other mechanisms. So it's important also to study microglia cells in slices, in vivo, and also in organotypic cultures. And here we can actually measure now the contact between microglia cells and, in this case, axons or dendrites. We can actually see live if microglia cells is eating away a synapse. We can now measure under certain conditions how much contact there is between neurons and microglia cells. For example, also a component of aging can be studied. We can do it with knockout or knock-in mice, and can actually see how much microglia activation can actually also destroy synaptic connections. Until the very end, even neurons might be killed. And one of the ways microglia cells in the brain can be activated is if there's an inflammation, an infection in our body. So it's not necessarily that the brain has to be infected. There are signaling mechanisms into the brain that can also activate microglia cells. And if this activation leads to chronic inflammation in the brain, that might actually contribute, or actually might increase the risk of becoming a neurodegenerative disease like Alzheimer's disease. And you can actually mimic this, I will come to that later, what is called a sterile injury or sterile infection, and we just use again cell wall components to study the process. And it has been known for a long time that there's a phenomenon which is called sickness behavior. This is related to when we become ill. We also have a certain type of behavior. We very often avoid social contact. We stay silent. Anxiety is increased. Also depressive-like symptoms are correlated to that. And there are a couple of papers. Here's one from the lab of Marco Prinz from Freiburg, who showed which signaling pathways from the immune system can actually have a direct effect on the nervous system, showing again how much the immune system can also shape neuronal function. There's another interesting thing which I think is important to highlight also if one wants to have an outreach of this scientific program. I've taken here a rather drastic picture that one of our immunologically largest organs is actually in belly fat. And there are many macrophages in there, and if you consider that obesity is now a worldwide problem, so there are more people on this planet obese than there are hungry people, and not because the situation for hungry people became better. So this is one of the important components to keep in mind, that obesity is a risk factor for Alzheimer's disease, a lot more than many other factors that have been discussed before. So lifestyle is influencing the incidence of Alzheimer's Alzheimer's disease as well. And my interpretation is that in addition of inducing diabetes, one of the things that happens is with obese people, especially when the belly fat is becoming very, very thick, that you have a constant production of cytokines, of inflammatory molecules, which might enter the brain and might activate microglia cells. And here, just to show, already in 2008 there was a publication in Neurology showing actually that if people had a high amount of belly fat, the risk to become Alzheimer's disease 30 years later, so many, many decades later, not just 10 years later, 30 years later, was highly increased, which can be metabolic functions, it can be the support of the brain with glucose, but it can also have an immunological function, because they do have, as I said before, a chronic neuroinflammation in their body going on, that in comparison to lean patients, if you have a lot of belly fat, you have a lot more production of cytokines. So which means here, highlighted in a review by the group of Michael Henniker, you see that there are lifestyle components in addition to genetic risk factors that actually influence the occurrence of getting Alzheimer's disease. And one is actually having a sepsis, poor oral health is another one, obesity is one, and reduced physical activity is another one. You should also know that physical activity increases BDNF levels also in the brain. So again, we have here a connection to neurotrophins that might be important in that respect as well. And the interesting thing is that for the microglia activation, you need a key molecule, a key complex. It's called the NALP3 complex. And this complex you can knock out in mice studies in order to see what happens if microglia cells cannot be activated anymore, because the microglia cells can then have consequences on other microglia cells, but also on astrocytes and other cells. They will increase the production of the nitric oxide synthase, which will then lead to the production of molecules which can be highly toxic for neurons and not only for pathogens. So what happens if one takes the NALP3 out of the system? The model mouse we have used together with the group of Michael Henniker with the APP-PS1 mice, where you had a knock-in of two human genes which have mutations that in humans lead to a high risk of becoming Alzheimer's disease, and in mice 5 to 6 months old they start to produce plaques. So in this model system you can partly replicate what happens in humans, and as Ed Kuh has highlighted so nicely in his talk, there's not a model system that perfectly fits the complete disease, but you can study in a reductionist approach aspects of the disease, and here you can study the aspects of what amyloid plaques might do to synaptic plasticity and the function. As you can see here, they are now plaques produced in the brain of mice which are not there under natural conditions, and also close to the plaques you will see microglia cells that are invading to these plaques. So they are highly activated in the human brain, but they are also highly activated in the mouse brain. So one of the studies we have done, and in principle you have seen part of that before, in APP-PS1 mice you see a reduced capacity to induce synaptic plasticity, as has been highlighted in the two talks before. But if we cross this APP-PS1 mice to the NALP3 knockout mice where the microglia cells cannot be activated anymore, we get normal LTP again, and also for components of structural plasticity. So as you have heard in the talks before, that not only mice but also humans lose synapses with the onset of the disease, and this can be prevented if we use NALP3 knockout mice. It should also be highlighted here that there's a knockout of another caspase, not the caspase 3 that Ed Kuh has talked about, this caspase 1 that's needed downstream after the activation of the NALP3 complex, that is important for microglia action connection when they are activated. If we knock that out, we get the same effect as with the NALP3 knockout, that we don't see any negative consequences on structural or functional plasticity. And also, that I find very important, the behavioral phenotype is completely rescued, because APP-PS1 mice are actually indeed better learners in comparison to wild-type mice at the same age. But if mice, APP-PS1 mice, are crossed to NALP3 knockout mice in a Morris water maze, they are trained to find the platform as fast as wild-type mice, whereas APP-PS1 mice are extremely bad learners, so they can extremely badly memorize a place where, for example, a hidden platform is maintained. We then went on and wanted to know if sepsis actually is a risk factor, and that there are some human studies indicating if people were in a hospital and had a sepsis, that the occurrence of mild cognitive impairment a couple of years later is highly increased. And you should know that mild cognitive impairment is a risk factor of becoming Alzheimer's disease. So the study we designed was very similar to the NALP3 knockout study I presented before. We basically wanted to induce a sepsis in a mouse model by injecting LPS component into the belly area, into the intraperitoneum, and wanted to see what happens to the brain actually. So it's a complete peripheral infection, not an infection, it's an injection of a bacterial cell wall component that mimics an infection, and that can actually lead to neuroinflammation in the brain. And the question is this then a risk factor for becoming neurodegenerative diseases? So we have used different cell wall components from E. coli and also from Salmonella typhimurium. We have waited 3 months so mice could completely recover this treatment. As you will see, they suffer quite a bit in terms of weight loss, but they recover quickly. And then we analyzed young mice at 7 months of age, or really old mice at 19 months of age. We studied again neuronal structure. We also studied neuronal function with synaptic plasticity and electrophysiological method, and we were again doing a behavioral assessment in addition to looking at microglia activation. And what you can see is in young mice, and that's kind of a nice control, that when we induce a sepsis, that they recover completely, also in terms of synaptic function of neuronal connection, and also synaptic plasticity is completely normal. So there are no changes in dendritic complexity or spine density, and the picture is quite different in old mice, where the morphology of neurons is significantly affected, so they lose branches. They also most likely lose synapses, because if we look for the postsynaptic site for spines of synapses, we see that spine density is highly significantly reduced after such a sepsis. So overall, there's a decrease in CA1 dendritic complexity in old animals, and that's also interesting, was more pronounced in the Salmonella LPS than in the E. coli LPS. So the immunological response of the periphery, which is larger in Salmonella, seemed to affect to a larger extent the chronic inflammation in the brain. We then did also synaptic plasticity experiments, and you see that in Salmonella typhimurium we see a decrease in LTP induction, whereas for E. coli we didn't see an effect, so cell wall components do count. But the interesting thing is that 3 months after the mice had recovered from a sepsis, we can still see functional and structural effects in old mice. So there is a component that actually mimics even some early components of the induction of Alzheimer's disease. So we see a reduction overall in LTP. Again, in old mice, if we look at behavioral experiments, you see that they have a harder time to find the quadrant if they had a sepsis before. Keep in mind, they completely recovered from the sepsis, but they still had a decrease in their learning, in their spatial learning capability. We have a mathematical way to actually calculate when mice or what kind of strategy mice use to find a platform. And the one we are interested in is the one that is hippocampus dependent. Same is actually for humans for spatial memory. And as you might know, the entorhinal cortex and the hippocampus are one of the first areas being affected with Alzheimer's disease. And here you see that after animals had sepsis, especially for typhimurium, you see this red area decreases, which means in comparison to control conditions that they use less hippocampus dependent strategy. They use more like floating around. So they can't memorize where the platform is, but they have a strategy to find it in the end, because they have learned if they float around that they will also at a certain stage accidentally hit the platform. Next thing was to look at microglia activation in old mice, and also here we see that there's a huge activation of microglia cells 3 months after the sepsis is gone. So the microglia cells stay active, and in the moment we are looking for epigenetic changes in the chromatin of these microglia cells to see why such a long-term response is maintained. Here we actually looked at the strength of the microglia response 24 hours after we injected the LPS, because we wanted to know if the initial immunological response is a predictor of the long-term consequences. And indeed, the stronger the initial immunological response, the longer microglia cells are activated in the end. So it could be that it's actually not the cell wall component or any direct effect, it could just be the response of the immune system that's actually determining the effects that are happening in the brain. So what I've shown to you so far is that we have age-dependent vulnerability towards and peripheral immune stimulation, a reduced dendritic complexity, a decrease in spine density, an increased density and activation status of microglia cells. We do see no changes actually in the number of astrocytes. Then we see that the LTP defect is a lot stronger in Salmonella typhimurium, and that they use, if they use learning strategies, they are less hippocampus dependent, so they also have learning deficits. So overall, LPS-mediated immune stimulation can induce long-lasting alteration in neuronal morphology and function. Then, coming back to the already introduced APP-PS1 mice, we also wanted to know how they react to an LPS injection, and actually there they even respond now to E. coli for morphological effects. So for the LPS that had a less high immunological response in the wild-type mice, in APP-PS1 mice, this response becomes larger, and it's also more long-lasting, and it's highly significant how there's a decrease in dendritic complexity, and also again in spine density, so synapses are affected as well. Same is true for LTP induction, which is now heavily reduced, which was not the case, as you might memorize, for the LPS E. coli that were added to wild-type mice. So we see a stronger effect overall, and again a decrease in LTP, and we see long-term effects of LPS on neuronal morphology and synaptic plasticity in adult and aged APP-PS1 mice. So as you can see here for LTP, and as you can see down here. And in addition, for looking at APP-PS1 mice and at wild-type mice, we again came back to the NALP3 knockout mice, because again, when we were doing the same LPS injections in NALP3 knockout mice, in APP-PS1 mice, then could prevent the effect. Here you can first see that we can prevent the effect under control mice if we cross if we have, instead of wild-type mice, NALP3 knockout mice, we can see that the effect of LPS of Salmonella is actually gone, which might actually really give a good indication that it's mediated via microglia glia cells. So in NALP3 knockout mice, we have normal morphology, normal LTP, and normal spine density. And actually, you might actually argue, well, if the NALP3 is a complete knockout, there might be developmental effects or effects via other cells. So we actually added acutely a blocker of the NALP3, the MCC 950, which we got from the lab from Michael Lutz from Bonn, and we see the same effect as in NALP3 knockout mice, indicating that the microglia cells are indeed contributing to that. So to summarize this sepsis model, we see an age-dependent sensitivity, which also fits the human phenotype. There's a microglia activation 3 months after immune stimulation. We see that LPS from Salmonella to Fumarium gets a lot stronger response, even in wild-type mice, than E. coli LPS. We see that part of the response, or probably the complete response, is mediated via the NALP3 inflammasome, and we see a stronger effect in aged APP PS1 mice, which might also mean, if this is at least in this sense a good model for the human Alzheimer patients, that becoming an infection in an aged individuum who is at risk of becoming Alzheimer's disease might be an additional risk factor that might be important here. So again, as I've indicated, we have now looked at sepsis, looking at the model that there might be different roads to increase the risk factors of becoming Alzheimer's disease, and an inflammation in the periphery might even be one of these risk factors, and the sepsis as being a huge activation of the immune system might be one. I will not have time to talk about our study, what we did with influenza virus type type A, because we also studied how mice reacted to different types of influenza viruses, because it has been known from the biggest flu pandemic in 1918, where probably more people died from the flu than actually in the war, that there are also neurological symptoms that have been described by Konstantin Vanikonomo that showed that something is also happening when you get the flu. So we were studying different strains of influenza viruses, which lead to a different response size. And again, the stronger the immunological response, the stronger is the phenotype we see in the nervous system. But here the important thing is that the H3N2 is just an infection of the periphery of the lung, whereas the H7N7 can actually enter the brain, so it's neurotrophic, not neurotrophic, so it can go into the brain and can have damage there, but the other virus, the H3N2, cannot, and we were looking at different phenotypes here. To cut a long story short, what we see is that neurotrophic and non-neurotrophic viruses can lead to effects in the brain and can lead to consequences in terms of neuronal morphology, of synaptic plasticity, and also of the learning behavior of the mice. Again, 3 months later, after they have been infected with influenza, they completely recover. The peripheral immune system is completely normal again, but microglia cells can stay activated for a long time. We have published that in Journal of Neuroscience more than a year ago, so I will not have time to go into detail here. I would just like to highlight that probably one of the risk factors one should keep in mind for getting Alzheimer's disease are also peripheral infections that might actually contribute via a large immune response and activation of microglia cells which stay activated. You might also ask which are the molecules that are doing the transmission between the immune system and the nervous system. And one of the receptor types and one of the signaling system is the interferon system. And the interferon receptor 1 is one of the important molecules also in the brain, being expressed on neurons, being expressed on astrocytes and microglia cells. And the signaling between these three cell types might also contribute either to the homeostatic brain of the brain to be physiologically active, or if there's a high expression or high release of interferons interferon receptor 1 system might actually also have negative consequences, which also highlights the fact that we have to take into account the amount of activation that will then lead to different effects. Our story, where we have knocked out the IFNA receptor in neurons, in astrocytes, in microglia cells, is that in that case not microglia cells but astrocytes are important for the receptor expression, and actually astrocytes also contribute to synaptic plasticity in a way that they remove glutamate from the synaptic cleft, and the more efficiently they do that, the more they can influence synaptic plasticity. And here the IFNA receptor system is leading to a different expression level of GLAST of one of these reporters. This is a huge story, that's a talk in itself. I just wanted to highlight that it's also important also for the aging brain to study is the connection and the interplay between the nervous system and the immune system, and the immune system and the nervous system, and their molecules that are involved here that are important, and the interferon system is just one of the components. We are astrocytes that might be important here. So this is again showing the complexity of what we need to deal with. And as you have heard before from the talk of Ed Koo, that the APP system is cleaved into different parts. And one of the pathways from the amyloid precursor protein leads actually to the production of A-beta. But there's another pathway that leads to the production of APPS-alpha. And we have invested a huge amount of work together with the lab of Ulrike Müller to actually show that APPS-alpha is neuroprotective and is increasing synaptic plasticity. So in addition to all the complex work you have heard today or you have read elsewhere, it might also be important to keep in mind that sometimes it would already help to promote a neuroprotective pathway of cleavage and to prevent a pathway that's at least a dangerous route that might lead to the production of A-beta, which can lead to oligomerization and to all the long-lasting effects. So the balance here might be important, and we have published papers showing that adding APPS-alpha can actually help to prevent, also in APPPS1 mice, the negative consequences. I will not go into details here because, again, this is a study in itself where we will not have time today to talk about it. But what I wanted to highlight is that when we were looking at microglia responses and responses of the immune system, our interpretation is not that this immunological response just comes into play after all the disaster that leads to Alzheimer's disease is already happening. We actually think that microglia cells are not just bystanders, they might actually promote the progression of the disease in the aged human brain, and that the immunological response of the brain is a risk factor. I don't believe in single golden bullets for the treatment of Alzheimer's disease, but it's probably important to keep all the complex interplay between A-beta and tau of synaptic plasticity in addition to immunological reactions. This is a point I wanted to make before. And our immunological reaction in the brain might also be affected by lifestyle factors. I've highlighted obesity, and actually many infections you can actually prevent if you get vaccination at the right time. So that might be also contributing factors that might help to reduce the risk of becoming Alzheimer's disease. These are the people I need to thank. We collaborate with Charles Dinarello in Colorado, with Michael Henniker in Bonn. We have a viral expert, Klaus Schugart, in Braunschweig, and Ulrich Kalinka is another expert in Braunschweig in Hannover as well. These are my people who did the work, and I thank you for your attention. Thank you very much.
In the initial part of your talk, you had mentioned about a study done in rats. When they go into the dark, they receive an electrical signal. And you also mentioned that those set of rats get killed, and you feed them to the naive rats. So I didn't understand, like, what did you actually...
Yeah, I will explain the experiment in more detail. So actually, rats were trained to avoid the dark. Naturally, if you give a rat a choice between a light area and a dark area, they go into the dark area. And they've learned to avoid the dark area because every time they went into the dark area, they got an electric shock. And when they have learned that, the assumption was there are changes in the brain of these rats, and probably the changes are stored in single molecules. Keep in mind the immunological concept, you do have B memory cells that produce antibodies that are specific for surface receptors of a specific subtype of a virus or of a bacteria. So there's a single molecule that has a memory about something it has experienced before in the immune system. The thinking in the nervous system was very similar. So it was actually thought that there's a single molecule, it was called scotophobin, might actually store the information of avoiding the dark. So the rats were killed after they have learned that, and the brain were actually fed to naive rats that have not experienced the learning situation before. And in the first publication of that experiment, it actually was claimed that they learned faster, indicating it might indeed be the case there might be single molecules storing memories. That's why the New York Times had the headline, "Eat your professor," because, okay. The story is most likely not true. First of all, the experiments could not that easily be repeated. Second, as you can imagine, professors did everything to prove the study wrong, just not to be eaten. And most likely they have not fed a protein to other rats, they have probably fed a stress hormone that was actually highly elevated, and if a rat is stressed, it's more alert and it might actually learn faster, but it has nothing to do with single molecules storing information. I think that was nicely explained in the talk of Dr. Sachikuma, who explained that for memory storage you need associative neuronal connections that change the network properties of these neuronal connections, and not just single sites, and for sure not single molecules that store information. Thanks for asking.